So the draft sequences were completed were published in 2001, and they proved to be extremely valuable right off the bat. For the first time, you had a complete collection of, or very nearly complete collection of all the protein coding genes in the human genome. You had very high quality draft sequence scaffolds along each chromosome. You had a much better indication of how repeats were organized within the human genome, how the different kinds of sequence elements were organized near the telomeres, the centromeres, and so forth. But there were still a tremendous number of gaps in the genome. While the draft was very useful, for example, for doing evolutionary studies of individual genes, you could take the human draft and compare it with the mouse draft and the model organism genomes. There were a lot of questions that could not be answered well with just the draft sequence. So the next phase of the Human Genome Project was creating the finished reference sequence, and this was a large, painstaking process. Going from draft sequence with lots of gaps to a finished sequence may not sound like that big of a deal, but it turns out that it's extremely tedious. You need to go into each gap that's there, usually on a back-by-back basis, figure out why there's a gap. Very often those sequences are particularly difficult to sequence. Sometimes they're virtually impossible to sequence, in which case you need to define the segment that can't be sequenced and flag it as not, as a certain size, and incorporate that into the finished sequence. Where there were gaps between clones, you needed to go back into back libraries. Any and all kinds of back libraries and YAC libraries were screened to try to find a piece of clone DNA that spanned a sequence gap that was between clones. And this went on basically chromosome by chromosome, back by back, for a couple of years until finally in 2003, a quote-unquote finished human genome sequence was declared. It still had some gaps, still had some problem areas, but all of the available clone libraries had been screened. All of the difficult regions had been attempted at least several times, and so at that point it was declared complete for now, and as technology improves we would try to improve it some more. So I'm going to go over that last finishing phase fairly briefly in this lecture. Following that, I'll introduce the next-gen sequencing methods that were introduced in 2005, 2006 primarily, and this was a very exciting, really revolutionary advance, primarily because it had the potential and has the potential and is continuing to show the potential to really drive down the cost of DNA sequencing to such a level that it's become one of the methods of choice for any kind of functional analysis having to do with the genome. And we'll get to that in a few moments. So we'll talk about finishing the human reference sequence, and in particular, how that really highlighted the prevalence and the importance of segmental duplications, copy number variations, and structural variations as important sources of human genetic variation, in addition to the single nucleotide polymorphisms. And as I mentioned before, we'll talk about the introduction of next generation sequencing technology. So finishing the human genome. So in both the Celera and the human drafts, as I mentioned before, we had high quality draft sequences. We knew where most of the sequences were along the chromosome, roughly speaking, but there are still a lot of pieces that were flipped around, some entire clones that were put in the wrong places. Gaps both within the assemblies of backs themselves and gaps between the contigs that had no clone coverage. So all these problems needed to be addressed, and they were addressed by the International Human Genome Sequencing Consortium, the public consortium. Celera worked primarily on annotation for a while, and then they faded out of the picture as the human reference sequence became the predominant workhorse. So the finishing process, as I mentioned, was clone by clone, chromosome by chromosome. But at the end of the day, the finished sequences were completed. And by comparing them with the draft sequence, you can get a sense of the kinds of problems that were solved. What you're looking at is a comparison of the finished sequence of a chromosome with the draft sequence, and the diagonal shows regions where they differ. And you can see a couple of regions where they differ a lot, where you can see clear chromosome segment inversions of chunks of draft sequence that stretch for megabases of DNA. So those were misoriented or somehow mislocalized contigs along the particular chromosome. Those were pretty significant differences. But even at the microscale, which is blown up in the small square at the right, you see even over the course of a few hundred kilobases, you can see a lot of mislocalized, misoriented draft fragments that were ultimately corrected by the finished sequence. And that turned out to be extremely important because in thinking about how one wishes to annotate a genome, if you're going to be looking for all of the genes and all of the splice variants, you really need to be confident about the linear order of the DNA sequence and sequence fragments along the chromosome. So the finished sequence had about 2.85 billion nucleotides. It encompassed at least 99% of the euchromatic human genome. So that's the part of the genome that does not include the centromeric heterochromatin and the high copy repeats, as well as the acrocentric short arm regions of the human genome, which were primarily tandem repeats and heterochromatin as well. In the sequence region, it had a very low error rate, 1 out of 100,000 bases. There are a total of 341 gaps. 33 correspond to the heterochromatic gaps that I just described. There are also 35 euchromatic boundary regions where the euchromatin is juxtaposed to the heterochromatin. And in these boundary regions, there were a lot of gaps as well, in part because the heterochromatin euchromatic chromatin is not a defined sharp boundary. There's sort of an interspersion of heterochromatic elements with euchromatic elements in these regions. And so they were particularly difficult to clone and assemble. There are 273 euchromatic interior gaps. So these are segments of what looks like fairly typical euchromatic DNA, where there's simply a gap there. And it turns out that about half of those interior gaps were associated with segmental duplications. And that helps to explain why the gaps were there right off the bat, because the segmental duplications are defined as stretches of DNA greater than a KB, greater than 90% nucleotide sequence identity. Some of these, in fact, a significant number, are much larger than that. They could stretch for tens or even hundreds of kilobases in size. And if you have a sequence of DNA of that size and that's present in multiple regions of the genome, it becomes very difficult to try to uniquely assemble any given copy of that duplication into the appropriate location in the genome. And so that helped to explain why these gaps were there. In addition, there were structural variations associated with these interior gaps. Now, the structural variations are defined as regions of significant insertions, deletions, or inversions relative to the reference genome. So what likely caused many of these gaps were differences in allelic structure from the different libraries that were used to create the initial human reference sequence. So if you have clone A from one allele, let's say from individual A, and you have clone B from individual B, and in clone A there's a 60 kilobase deletion relative to individual B, there's no way that those two clones are going to be able to be assembled properly. And so what you'll end up with is a gap in the sequence. And so many of the remaining interior gaps were simply differences in haplotype structure that were not simply single nucleotide polymorphisms, but due to significantly sized duplications, deletions, or even inverted regions of chromosome sequences. Finally, when looking at the finished sequence, the protein encoding G number actually went down even farther. So the annotation of the finished sequence resulted in a number of about 20,000 to 25,000 protein encoding genes, which is what the estimate remains right now as far as protein encoding. But as you'll hear in other lectures, there's been a huge explosion in the number of transcripts that have been identified, and in particularly the long non-coding transcripts. So in comparing the finished with the draft, as I mentioned, 341 gaps in the finished, there were about 150,000 gaps in the draft. The continuity is measured in the N50 contig size, went from about 38 kilobases to about 80 kilobases. And in fact, for some chromosomes, almost the entire chromosome is present in two or three very large contigs with just a couple of gaps. In the finishing process, the orientation and order of draft contigs were corrected, as I mentioned. And also a significant source of error was actually artifactual sequence duplications. It turns out that because some of the quality of the sequence in the draft was subpar, what was left in the actual draft was a piece of assembled sequence that was high quality, a piece of assembled sequence that was actually from the same region but lower quality, and so it failed to assemble properly. And so you actually had these sequence duplications sort of embedded in the draft that once the sequence was completed and the lower quality material was removed, you were able to eliminate those artifactual duplications. So how did we know the sequence was good? There are a couple of quality control steps. Fairly significant effort was put forward to really examine the finished sequence and try to figure out how good it was, where the problems were, and what might be done to perhaps improve it in the future. In the first instance, a number of backs were randomly selected and sent to a group of sequence finishers that were not involved in the actual construction of the original finished sequence. And they were resequenced to very high redundancy, and to compare and look for sequencing errors and a sequencing error rate. And it turns out that those error rates were extremely low. 1 out of 100,000 were less than 50 base pair events, and even smaller fraction were greater than 50 base pair events. And most of those errors that were greater than 50 bases corresponded to tandem repeat errors. So those are regions of the back where you had multiple copies of the identical sequence, and you may have had a fewer number or a greater number of copies of the tandem repeat reported as were actually present. So these are fairly minor kinds of errors. By looking at the regions of the finished genome where the clones overlapped, you could look at the SNPs in the region, but also look at the tandem repeats in the region and ask the question, how many errors occurred within the tandem repeats of the overlap region. And overall, from both the independent resequencing and the clone overlap analysis, the total error rate was extremely low, about 20 to 100-fold lower than the natural polymorphism rate. So that gave investigators a lot of confidence that this reference genome could be used very effectively to look at not only genome structure and gene content and so on and so forth, but also at variation between additional genomes and this reference sequence. In addition to these quality control steps, a phosmid library was created from an individual genome, and all of the phosmids in the library were sequenced at both ends. And this gave the community a set of paired end sequences of a particular orientation that covered the entire genome about 8 times. And by mapping each of those end sequences back to the reference sequence, you could ask the question for that individual phosmid, is the expected distance there between the two end sequences, and are they in the proper orientation? And it turns out that both end sequences were in the proper orientation and distance for the vast majority. And where there were differences, in some cases it was due to error, but in many cases it was actually due to polymorphism between the genome from which the phosmids were prepared and the reference genome. So by using these different quality control metrics, it could be determined that the finished sequence, while not perfect, was certainly of extremely high quality and could be used for variation analysis. As far as coverage, greater than 99% of the euchromatin appeared to be covered in this genome. And if you looked at all of the cDNA sequences that were prepared from all of the various EST projects and cDNA sequencing projects, which were done independently of the genomic sequence, and you looked for those cDNAs in the genome, the vast majority, 99.74% of the cDNAs aligned to sequence, with a fairly small fraction, about 0.5%, aligning to more than one location, indicating that some of the cDNAs were derived from regions that were duplicated within the genome. A very small percentage had a high nucleotide sequence divergence from the genome sequence, and many of these corresponded to regions of the genome that were already known to be fairly variable from individual to individual, such as the major histocompatibility complex region and the immunoglobulin region. The remaining cDNAs, those that didn't map completely, a very small percentage, 0.06%, were completely absent. So they must have been present in gaps in the finished sequence. A slightly higher percentage was partially absent, and these were primarily adjacent to gaps, but not all of them. Some of them appeared to be polymorphisms or perhaps errors in the current sequence, and that was still left kind of ambiguous. In addition, a random plasmid end sequencing was done for a large number of small plasmids. Once again, the end sequences provided expected orientations and expected spacing between orientations, and when all of these alignments were done, greater than 99% of the end sequences mapped as expected. There were a very small fraction where neither end aligned, suggesting that the entire small fragment was missing from the reference sequence, which could mean either a polymorphism from between the genome from which the plasmids were prepared and the reference sequence, or it could mean that the plasmids actually fell within one of the remaining gaps in the reference sequence. So I mentioned segmental duplications a couple of times. There turned out to be a bit of a surprise in that we always knew from the earliest days of the Human Genome Project that there were some regions of the genome that were duplicated, but the extent of segmental duplication was not anticipated. Now, these are chunks of euchromatic DNA that are basically lifted up and duplicated and put in another region of the genome, and it can be in 2 copies, 5 copies, 10 copies, even greater numbers of copies. It turns out that they're highly enriched in the pericentromeres, which are the regions right next to the centromere, and also the subtelomeres. So these euchromatic boundary regions had an elevated level of these segmental duplications, suggesting perhaps that these regions of the genome are slightly more rapidly evolving and more malleable than other regions of the euchromatin. And also the segmental duplications were associated with large polymorphisms. So different individuals may have different quantities of these segmental duplications at a given location. So this led to the term copy number variation, which means you have, in one individual, you have a certain number of copies of a segmentally duplicated piece of DNA, and another individual, you have a different number of copies, either at the same locus or perhaps additional loci within the genome. And this is a kind of variation that hadn't really been anticipated and thought about very much. But it turns out that the structure of these segmental duplications sometimes gives rise to susceptibilities to deletions associated with these segmental duplications. And those turned out to actually be important for the genesis of certain diseases. So particularly some multigenic diseases where you have chromosomal deletion events or chromosome duplication events, very often they're flanked by segmental duplications, which leads to a possible mechanistic explanation for why these regions are more prone to deletion and duplication. So this is a figure from the finished sequence of chromosome 7 that came out in 2003, highlighting the segmental duplication content. Chromosome 7 is shown in the center of the top of the figure, from the P arm on the left to the Q arm on the right, and the regions that are duplicated are shown in both the blue and in the red line segments. The blue segments connect segmental duplications that are present within chromosome 7. So wherever you see a blue segment poking up, it connects a segmental duplication within chromosome 7 that's present in multiple sites. Where you see the red lines going to the other chromosomes that are outlined above and below the chromosome 7, you see interchromosomal segmental duplications. So these are chunks of chromosome 7 that are present at multiple sites on other chromosomes. So you can see that a fair amount of territory, chromosomal territory on chromosome 7 is actually comprised of these segmental duplications. And it turns out that genome-wide, roughly 5% of all of the euchromatin in the human genome is segmentally duplicated, which is a much larger amount than what had been anticipated. So what's in these segmental duplications? It turns out that many of them correspond to gene families that have been characterized, and you can see a sampling here of some of the genes. Some are familiar players: olfactory receptors, immunoglobulins, interferons. Many of these have properties, have the property that they're fairly rapidly evolving. They interact with the environment, like receptors, olfactory receptors, immunoglobulin receptors, they're involved in functioning of the immune system. So these kinds of genes that encode proteins that interact with the environment may be especially susceptible to being embedded in this segmentally duplicated DNA. And there may be a biological consequence in that, because they're highly variable from individual to individual, that variation can be acted upon by natural selection, and it may lead to greater fitness for the species as a whole to have these kinds of functions redundant and rapidly changing within the population. I like this figure. This really illustrates what I think is one of the key important aspects of having a finished sequence. You see on the top the finished sequence of human chromosome 16, and on the bottom you see the whole genome sequence assembly for chromosome 16. And in the middle you see a segmentally duplicated segment of DNA that was all completely collapsed into a single tiny locus in the whole genome shotgun assembly, but clearly present in many locations just within chromosome 16 in the finished sequence. And this illustrates what many people see as a major disadvantage of whole genome shotgun assemblies. Even high quality, highly redundant whole genome shotgun assemblies tend to overcollapse these highly similar segmental duplications. So you get an incorrect and inaccurate picture of how the DNA is actually organized along the chromosome in the shotgun assemblies. Nonetheless, because of the difficulty in creating the finished sequences and the level of effort and expense that goes into it, even now the only truly finished sequences are the human sequence and the mouse sequence for large genomes. For model genomes, it's a different story. They have lower repeat content, so many of the model genomes, including the nematode, yeast, Drosophila, they all have finished sequences as well. But for large genomes, they're all still basically high-quality drafts. One other thing I should mention before I move on to the next generation sequencing technologies in particular, a key value of the finished sequence really became evident when the next generation sequencing technologies were developed, in that you need a high-quality reference sequence in order to map these short reads that are generated by next generation sequencing technologies back to a reference sequence, and you'll see that in a moment. Before I get to that, a little bit more about structural variation. I talked about segmental duplication content and how that affects the assembly and the gene content and so forth. Structural variation refers to any large duplication, deletion, inversion in a genome relative to a reference genome. And these can be detected readily by looking at fragment mate pair mappings. If you map both ends of a DNA fragment, and you know the size of the DNA fragment, then you can predict where the reads should map in and what orientation. If they violate either the distance requirement, that suggests there's either an insertion or deletion, or the orientation requirement, which suggests that there's an inversion of the source genome relative to the reference genome. And these kinds of changes are referred to as structural variations. And once again, similar to the segmentally duplicated and copy number variant regions, this was a relatively underappreciated source of variation in the human genome. And it turns out to be quite important. Structural variants can be generated by a number of mechanisms. This is one potential mechanism that's certainly at play, where you have segmentally duplicated DNA fragments of different sizes. They can recombine meiotically and generate both deletions and duplications. So this is one way that you can generate structural variants. Other ways are not clear. Perhaps genomically, meiotically unstable regions may have a greater propensity to generate structural variants. Certain kinds of DNA sequence elements might give rise to structural variants more frequently than others, and so forth. But it's still not clear how many forms of structural variation are arrived at. So there was a key paper that was published in 2005, actually in Nature Genetics. There's a mistake here, where the phosmid mate pairs that I described earlier that were used to test the validity of the human reference sequence were actually looked at more closely, particularly those that failed to provide validation of the reference sequence. And it turns out that many of those that failed to validate the reference sequence were due to these structural variations that I just described. And it turns out that many of the structural variations were associated, about half of them were associated with segmental duplications. And you can see that there were, in this particular data set, there were 52 that were deletions, 68 that were insertions, and 43 that were inversions that were associated very closely with segmental duplications. And these either occurred in one of the two chromosomes of the source DNA or on both of the chromosomes. So you had actually entire homozygous deletions in some cases, and homozygous insertions, as well as heterozygous insertions and deletions. And the human-specific repeats were also a major source of these structural variants, and these are typically human-specific transposons. So you had either the insertion or the popping out of a human-specific transposon that led to many of these structural variants. And some of them just occurred in unique sequence for unknown reasons. So this turned out to be a significant source of variation in the human genome. And in many cases, it covered not just a single gene, but in some cases many genes. So some of the validated polymorphisms carried along with them genes of known function, and these are listed here. So now in around 2005, 2006, a new type of sequencing was developed called next generation sequencing. And it's had a huge impact on genomics and tremendously expanded the scope of genome analysis, primarily because it's enabled the development of much cheaper and high throughput sequencing methods. So now DNA sequence can really be used as an analysis tool for all kinds of biological experiments. So some of the key differences between the conventional Sanger-type sequencing, which I described earlier, and the next-gen methods are shown here. You start out the same way. You fragment the DNA, but in the conventional method, you have to go through a cloning vehicle and a cycle sequencing type of step, as well as a gel electrophoresis step to come up with the DNA sequence. With high throughput sequencing or next-gen sequencing, you take the DNA fragments, ligate adapters, and then use those adapters to create single molecule libraries at a very high density on a variety of different formats. So in this case, they show a Polyne sequencing format. So each of the individual molecules can be made with an adapter that then sticks to a surface, and you can PCR amplify that single DNA molecule in situ on that surface, so you end up with a cluster of identical molecules, all based on that original single DNA molecule. And then you can use cyclic array sequencing to determine the sequence of that cluster of molecules on a base-by-base basis. So this is illustrated in the bottom part of the panel, where each individual spot can be imaged after each cycle, and as each spot is imaged, an additional nucleotide is added that corresponds to the next base in the sequence. And so you can, instead of having to run an electrophoretic gel to determine the sequence, here you can determine the sequence just by looking at optically the addition of fluorescently labeled nucleotides to each individual spot. So this allows you to tremendously compress the space required to do the sequencing, causing enabling a huge conservation of supplies and resources, in greatly expanding the number of sequences that can be acquired at one time. And so you have this massively parallel method of generating DNA sequence. So that's all terrific. The downside is the methods, at least initially, could only generate sequence of maybe 30 bases, 35 bases for each individual DNA strand. So while you could generate a huge number of sequences, they were all relatively short sequences. So the initial applications of next-gen sequencing were focused on those applications where you could make use of these relatively short sequence tags. Now, a key step in the next-gen sequencing method was the clonal amplification of single molecules. As I mentioned, you start by fragmenting the DNA and putting on linkers, synthetic linkers. And in some formats, you stick those to a solid support and then amplify them. And in other formats, you attach those to beads and then use what's called emulsion PCR, which is a mixture of oil and buffer that causes the formation of small spheres of aqueous solution. And within those spheres, you carry out thermal cycling and generate from the beads that have the single molecules attached identical copies of your single DNA molecule. And the emulsion PCR method is the method for carrying out this clonal amplification of single molecules for the 454 method of sequencing and the solid method of sequencing that I'll get to in a moment. The solid substrate based PCR was used very effectively initially by Solexa, which was then bought out by Illumina, to create multiple copies of single molecules using a method called bridge PCR. And here, the single molecules with the ligated adapters anneal to their complementary oligonucleotide strands that are on the surface support, and you use the reverse reaction to create a double-stranded molecule, which you then go through a series of PCR cycles to generate ultimately a large cluster of DNA molecules that are identical to the initial single DNA molecule at that physical location on the solid substrate. And this so-called bridge PCR became the method of choice for preparing clonal single molecule amplification for Illumina sequencing. So the cyclic array sequencing can be carried out by different flavors of techniques. In the 454 sequencing technology, the beads that were created and amplified within the emulsion PCR were then transferred to a specialized support that was formed of an array of small receptacles, each of which could only take in a single bead. So because of the relative sizes, only a single bead could fit into each one of these little compartments. And once you had all of the beads in the compartment, you could then do sequencing on all of the beads at the same time. And the way that was done is that different reagents were flowed over the top of the beads, carrying out a different step of the cyclic array sequencing. And for the 454, it was done in a fashion where a single nucleotide was flowed at each step. So you had a step where the A nucleotide was flowed over, then the C, then the T, then the G, and then so on and so forth. The key is that with each incorporation, when they used a modified nucleotide, they used modified nucleotides such that when the incorporation occurred, you had the release of a pulse of light. And this pulse of light indicated that at that particular bead, a nucleotide of a given type was incorporated at that site. And so by going through ACTG, ACTG, you could build up for each bead a DNA sequence corresponding to the sequence that was present on that bead. The cyclic array sequencing in the Solexa Illumina format was done in a different fashion. In this case, you have the bridge PCR products on a solid support. And at each step, you flow a set of reagents that includes each of the four nucleotides, with each of the nucleotides labeled with a different color. And depending on the sequence of each individual clonally amplified single DNA molecule, an individual nucleotide is incorporated based on the complementary base. So at each step, you incorporate either an A, a C, a T, or a G, and that's indicated by the color at that particular spot on that particular slide. And so you image after each cycle, and depending on the color that you get, you deduce the nucleotide that's been incorporated. Then you go through a series of chemical steps that leads to the start of a second cycle where you do the same thing. And here you add the next nucleotide on that growing strand and image that. And then you add a third cycle where you add an additional nucleotide. And based on the sequence of colors at a particular site on your solid substrate, you deduce the sequential addition of nucleotides that corresponds to the sequence on the clonally amplified single DNA molecule. And by doing that in a massively parallel fashion, you get a tremendous amount of sequence at the same time from these growing strands of DNA sequence that you're imaging. Once you have your 36 bases initially of sequence from each of those, then you can computationally align those back to the human reference sequence and do an analysis of whatever source genome that you in fact have sequenced. Cyclic array sequencing using the SOLID technology is based on a slightly different principle, and this principle is the identity of dinucleotides in libraries of small oligonucleotides. And each dinucleotide is labeled with a particular color, and based on the ligation of those libraries to a primer, which is located at defined distances that are in different registries that are attached to the clonally amplified DNA molecule. When you get the ligation, it means that you have that dinucleotide at a given defined distance from the primer oligonucleotide. And by doing this repetitively for multiple oligonucleotide libraries with defined dinucleotides, you can build up a very accurate DNA sequence of the DNA strand. Some newer variations of next-gen sequencing methods are shown here. Complete Genomics developed a novel way to create single molecule, clonally amplified single molecule populations using rolling circle amplification, and these so-called nanoballs can be attached to a substrate and sequenced, in this case, using technology very similar to the SOLID technology, using sets of oligonucleotides to do the sequencing. And this can be made extremely efficient. Ion Torrent is yet another technology that's come online fairly recently. It uses a principle very similar to the 454 principle, where you flow individual nucleotides over your surface. Each well of the surface contains a clonally amplified DNA molecule. In this case, the incorporation of a nucleotide is detected not by a pulse of light, but by the release of a proton that occurs when the nucleotide is incorporated. And so you have basically an extremely tiny pH meter in each of these positions in this wafer, and the detection of a slight pH change corresponding to the release of a proton corresponds to the incorporation of that particular nucleotide. And because the technology used to make these wafers is very similar to making semiconductors, these can be mass produced in a very cheap fashion, and enabling a potential to drive down the costs of doing this kind of sequencing. So I wanted to go into slightly more depth with 454 sequencing. That was the first major next-gen technology to come online. Some of its features are shown here. You can do sequencing by synthesis, as I mentioned before. You can get long reads with 454, which is really the most unique aspect of 454 sequencing. And because of the longer reads, it became and is still being used in situations where having relatively long reads is actually very important for doing the analysis of the sequencings. Getting mate pair information from your library source is possible using 454. The error rate is a potential problem. There are some kinds of sequences that it doesn't handle particularly well, so high redundancy is sometimes necessary to get high accuracy with 454. And the reagent costs are slightly higher than the other platforms. so that has also limited the applicability of the 454 sequencing. Nonetheless, you get a huge number of sort of moderate length reads. Actually, with the newer technology, you get up to 800 bases of sequence, and sometimes even more than that. But it turns out to be a very effective way to get molecules to get sequence information of this length on your source DNA. So again, a review of the technology itself. This is an emulsion PCR-based method, which is, depending on who you talk to, it's less convenient. It's more finicky to do emulsion PCRs than doing the substrate-based bridge PCR method, which is also limited to some extent the wide acceptance of 454 sequencing technology. You do the sequencing by synthesis, or the cyclic array sequencing, and you can get your reads directly off of the instrument in a massively parallel fashion. What you get data-wise is what's called electropherogram of the incorporations of each base along the DNA. And when you have multiple bases in a row, you get additional signals at exactly that same location. And so the number of multiple bases that happen in a row is reflected in a higher intensity of signal at that particular base position. The difficulty is that as you get multiple nucleotides in a row, it becomes a little more difficult to determine whether you have 2 versus 3 or 5 versus 6, or especially 7 versus 8 or 9 versus 10. And so you very often get errors in regions of sequence where you have these polynucleotide stretches when you're using the 454 sequencing method. So to summarize, 454 reads are longer, but they're lower throughput, and because of the polynucleotides, you have a higher frequency of error in certain kinds of stretches of DNA that have these kinds of repetitive structures. Illumina sequencing has become the predominant next-gen method, and both because of the convenience and also because of improvements in technology that have enabled extremely high throughput, have enabled mate pair sequencing possibilities, and have extended the bases from the original 30 to 35 base read length to now it's about 110 bases per read possible. The current generation of instruments is called the HiSeq, and you can see some of the statistics here depending on the read length that you want and whether you want mate pairs or not. It takes variable amounts of time to produce the data, but look at the amount of data that's generated in total gigabases of DNA. A huge number of bases of DNA are produced with each run because of the massively parallel nature of the HiSeq. So the flow cell itself can be divided into eight lanes, and so you can load a separate sample on each lane. There are ways to multiplex, so if you have a need for a fewer number of reads for a given sample, you can put on different kinds of adapters when you prepare your library, and then combine all of those samples into a single flow cell and get multiplexed information based on the unique tags that you've added to your sample. But the basic steps are the same as I showed you before in cycle sequencing. So you add your oligonucleotides to the fragmented DNA. You do the cluster PCR to generate your single molecule amplifications. And what you end up with are thousands of of DNA molecules identical to the single molecule, all localized physically within a single cluster on your substrate. And much of the technology improvement that's happened with the Illumina sequencing has been an improvement in both the chemistry and the optics in analyzing these the fluorescent signals from these clusters, so that you can separate out signals that are from clusters that are very close to each other. So you can pack more single DNA molecules onto a single slide and better discriminate between individual clusters. As I mentioned before, you grow the DNA strand by cyclic array sequencing. At each step, you image the nucleotide that's been incorporated at each spot, and because each nucleotide has been labeled with a different color. And with each growing strand on each of the thousands and thousands of clusters on a given slide, you image it between each reedition, and you get your DNA strand, which you can then map back to the reference genome. Now, the analysis is not straightforward, although there have been tremendous improvements in doing the alignments over the last several years. You start out with your image, to do the image analysis, convert that into bases, and each base has a quality value associated with it. And then the reads are aligned to the reference genome by various kinds of algorithms that can be used. And the alignment of reads to the genome can be used to identify initially single nucleotide polymorphisms or small changes in the bases, which is shown here. You have a two-base deletion a two-base pair insertion in your source genome relative to the reference sequence, as shown here. So some features of Illumina reads. The accuracy of the read drops off slightly at the ends of the reads. So there's a sweet spot where you have a very high stretch of accurate bases in a row in the center of the Illumina reads. At either end, it's still high quality, but there are more errors towards the end. So that has to be taken into account. The short reads limit the mappability in repeat-rich and segmentally duplicated parts of the reference sequence. Initially, especially with the 30-base, 36-base sequences, that's a very significant problem. Although you get a lot of information, you're still missing the ability to get information from many parts of the genome. With the extension of the reads to 110 bases, that helps quite a lot, but it's still significantly shorter than what you get from Sanger-type reads, which were typically 500 to 800 bases, and also the 454 type reads, which are also 500 to 800 bases. So you are somewhat limited in mapping back to the reference genome, and there are parts of the genome that can't be analyzed particularly well using these short reads. You can do paired ends with Illumina, and that's been a huge bonus, both in improving the mapping accuracy and also enabling some aspects of whole genome assembly. Although even with 110 bases, the genome assemblies are pretty poor, and it's very difficult to get good whole genome assemblies just using any next-gen sequence system all by itself. Typically, you have to combine it with longer read approaches in order to get whole genome assemblies. Coverage of the genome in Illumina reads in regions that are very high or very low in GC content is sometimes problematic. And this is likely due to the PCR steps in cluster generation and library prep. The polymerases used for PCR typically don't handle, under standard conditions at least, very high GC or very low GC in a consistent fashion. And so those sequences can sometimes drop out and be underrepresented in Illumina libraries. There have been improvements in this area. There are specialized methods that have been incorporated that reduce this kind of bias, but it's still there. So in addition to 454 and Illumina sequencing, the other kinds of sequencing using the nanoball type sequencing is also on the market. It's being used extensively. There are third-generation sequencing technologies that have been developed more recently. Some actually have instruments in laboratories now that are being tested and actually used to generate data, such as the PacBio system. And the advantages are that this is a single molecule sequencing technique done in real time. It generates very long reads, 3,000 to up to 10,000 bases. The disadvantage is that it's a relatively low accuracy, on the order of 85% accuracy. So while you can get longer sequences, you still need a very high level of redundancy in order to improve your accuracy level. Nanopore single molecule sequencing is still in the developmental phases. An instrument is supposed to be coming out relatively shortly from Oxford Nanopore. These are methods where an individual DNA molecule is drawn through a small pore in a membrane of some sort, and as it passes through, sensors detect the base that's present on the DNA molecule. Like the PacBio method, it can be done on single molecules, has the potential, tremendous potential, for improving the length of the sequence read, but they're still struggling, as PacBio is, with the accuracy issue. So if they can overcome those issues, these third-generation sequencers have a tremendous amount of potential for improving further the efficiency of high-throughput sequencing.